Hello everyone, welcome back to our YouTube channel Agri Addict. I am your mentor Ariprasad SK, undergraduate from University of Agriculture Science Dharwad, Masters at Indian Agriculture Research Institute in the Department of Seed Science and Technology, and I am currently pursuing my second year PhD in the same department in the Indian Agriculture Research Institute IIR in New Delhi. So I have secured All India 40th rank in my ICAR JRF exam, All India second rank in my ICAR SRF exam, and I also cleared IBPS A4 mains, and recently I have cleared the ASRB net in the subject Seed Science and Technology. So today in this video we are going to discuss about the Hardy-Weinberg law or the principle. So this is one of the most important principles or the law when you study this population genetics, right? So whatever questions that will be asked in your ICAR, JRF or SRF exam, or you can take the CUET or any other MS state entrance exams. So this concept is very much important where you can expect some questions from this chapter. Okay, so in the previous video we have discussed about the introduction to this population genetics, and next I have discussed about the population, like what is population, what are genes, and what is gene pool, and next I have discussed about how to calculate this gene frequency and the genotypic frequency. And next I have introduced you to the topic or the principle of this Hardy-Weinberg, and also given the equation, which is p square plus 2pq plus q square is equal to 1, and we have clearly discussed how it is equal to 1 and how this equation was derived. So if you have missed that video, please go back and watch that video before watching this present video. Okay, let us just start our discussion with the Hardy-Weinberg principle. So the today's content is like I'll be discussing about what is Hardy-Weinberg principle and what is this equilibrium. Like we might be regularly hearing this term where we say that they say like population is in equilibrium, whether the population is in equilibrium or not. So all of these kind of statements will understand like what does this equilibrium mean, and then we can understand those statements. Next I will also give you the proof for maintaining this, how the population maintains its equilibrium in case of random mating population, or generally how the population in the nature, if they satisfy certain condition, how the equilibrium in this population is maintained. So that I'm going to give you the proof. Next we'll discuss about certain assumptions that are needed, or certain assumptions that are made in order to work this Hardy-Weinberg law. Like in order to apply this Hardy-Weinberg law, there are certain assumptions that we need to take. So we'll discuss what are those assumptions in detail. Next or lastly, we'll discuss about the significance or the practical implications of this Hardy-Weinberg principle. Okay, so let us start with Hardy-Weinberg law. Okay, so see, the Hardy-Weinberg law states that the gene frequency and the genotypic frequency, so these are the two important parameters when we are studying the population genetics. So this law or the principle says that these two frequency will remain constant from generation to generation. That is what this law says. So this law states that the gene and the genotypic frequency remains constant from generation to generation with certain conditions or with certain assumptions. For example, if we have certain population, if it obeys certain rules, then the gene and the genotypic frequency will be constant from generation to generation. That is what this Hardy-Weinberg law says. So what are those assumptions? Like if a population is sufficiently large enough, and secondly if it is completely random mating, and third if there are no evolutionary forces which are acting on the population. Like what are the evolutionary forces which I am talking about? So the evolutionary forces can be mutation, it can be migration, or it can be selection, or it can be the random drift, or which is also called as genetic drift. So whenever there is a population which is sufficiently large, or which overall satisfies these conditions, so those population in those population, the gene frequency and the genotypic frequency, both of them will remain constant from generation to generation. That is what this law tells us. For example, if I have a G0 generation, so this is the base generation. So in this generation, if I derive the allelic frequency of a capital A and small a, so we are considering a single gene, which is, let us take the A is a single gene which we are studying, and it has two different alleles. So when it has two different alleles, so this we can calculate the frequency of a capital A and small a if we have the total number of individuals in the population, right? So in this case, whatever might be the value of this f of a, let us take it as p, it is p and it is q. So this value, whenever this population goes to the next generation, that is g1, the same, like when again if we are calculating the allelic frequency from this generation, that will be always equal to p, and the small a will be always equal to q when a population obeys these rules or it satisfies these conditions. At that time the allelic frequency will remain constant to from generation to generation. That is what you're seeing here. In the G0 generation, the f of capital A was p, and also in the G1 generation, f of capital A is again p. Again, if this generation goes to the next generation, again if the f of a or the f of small a is calculated here, it should be again equal to p and q. So this is in terms of gene frequency, or it is also called as allelic frequency. Similarly, so this was only one case. Similarly, if we are considering the genotypic frequency, so in the first place, in the G0 generation, if I want to calculate the f of capital A, what is this? So this is the genotypic frequency of capital A and capital A. And what are the different possible genotypes from a single gene with the two alleles? So it might be capital A small a, or small a, right? So if I want to calculate, or if I calculate this genotypic frequency of capital A small a, small a in the G0 generation, which I will give it as p square, and this would be 2pq, and this is q square. So these values what we are getting in the G0 generation, that should be same even in the G2, G3 and nth generation when it satisfies the above conditions which were discussed in the previous slide. So even in the G1 generation, the f of capital A should be again p square, f of capital A small a should be again q square. So that is what it means. I hope this is clear. So this is about the Hardy-Weinberg principle. Okay. So next, what is this equilibrium? So equilibrium, what exactly the equilibrium is? So I've just now explained you about the gene and the genotypic frequency remains constant from generation to generation. If any population satisfies this condition, any population satisfies these conditions. So what are the conditions? The conditions are like gene or the allelic frequency, it is remaining constant from generation to generation. First point. Second, the genotypic frequency, they are also remaining constant from generation to generation. If it satisfies this, or if any population satisfies this two cases, then we can call that population is in equilibrium. That means the population is stabilized, where you are not seeing any change in the gene frequency or the genotypic frequency. How this equilibrium is achieved? This equilibrium is because of no external disturbance. External disturbance in terms of what? External disturbance in terms of evolutionary forces. So there are no evolutionary forces which are acting on the population. That is why the allelic frequency and the genotypic frequency are not getting disturbed. They are maintained constant from generation to generation. So that is what the Hardy-Weinberg law or the principle says. So along with that law, we have that equation p square plus 2pq plus q square is equal to 1. So using this equation, also p plus q is equal to 1. So using this equation, we can easily calculate the gene frequency if we have the genotypic frequency. Like if the genotypic frequency is given, then based on the genotypic frequency, we can calculate the gene frequency. And if the gene frequency is already given, we can easily calculate the genotypic frequency just by applying this equation or the formula. So this is the practical implication of this Hardy-Weinberg law. So we are anyhow going to discuss these practical implications in the upcoming slide. So this is about basic information which is very much necessary for you people to understand all the other concepts in this population genetics. Okay, so next. So this gives. Now let us discuss how this equilibrium is maintained. So let us assume certain condition. So what were the conditions previously I said to be in the equilibrium? One is random mating. The population should be in undergoing random mating, and second one, it should be sufficiently large, and third one is it should no evolutionary forces are acting on the population. So let us assume for an instance that so all these conditions are satisfied. So when all these conditions are satisfied, I will show you like how this f of a in the g naught generation is equal to p, and also how this f of a in the g1 generation is equal to q square, p square. Both how they are equal and how they are maintained constant from generation to generation, that we can discuss. So now in this one we'll discuss about gene frequency first. So this was genotypic frequency. We'll just first discuss how this f of a in g naught is equal to f of a in the g1 generation. Okay, so let us assume a certain gene which is capital A. So this is in the base population which is g naught. So this capital A has two different alleles, let us take, which are capital A, capital A small a, and small a small a. And this is the genotypes which is possible from these alleles. So what will be the frequency of these genotypes? So this genotype frequency will be p squared, 2pq, and q squared. So what will be the allelic frequency then? Allelic frequency will be p and q. So this was in a G naught generation. Assume it is undergoing completely a random mating, right? So randomly they are fusing with each other. Then according to next in the G1 generation, what type of genotypes are formed? That purely depends on the alleles, right? So if I take this P and Q on the both the side, since it is completely random and you have every gamete has an equal chance to survive, so I can make this table where I can draw males on one side and females on other side, right? In that case, wait, so this is, so females on this side and males on this side. So if we cross, so these are the possible genotypic frequencies that we are getting in the G1 generation. Okay, so now we have got this genotypic frequency of f of capital A, f of capital A small a, and f of small a small a. So then what we can do? So from this genotypic frequency, we can calculate the allelic frequency, that is f of a and f of small a, right? From the which population? Which is the G1 population. So now whatever we will calculate, like if we are calculating the f of capital A, that should be again equal to p, which was there in the previous generation. So we have to now prove that f of a in the first generation is equal to p. That is what we have to prove now. So let us just prove that. So I'll take this values. Okay, for example, if I want to calculate f of capital A, so what it is equal to? It should be equal to number of AA individuals plus half into number of heterozygous individuals. So that will give us the frequency of capital A and frequency of capital A, right? So what if I just substitute the values? For example, what is the frequency of capital A? So frequency of capital A is p square. So what will be the frequency of capital A small a? It is 2pq, and you just substitute the 2 here. So in this case it is p square plus pq. So if I take p as a common, like it would be p into p plus q. So in this case we already know that p plus q is equal to 1. So if we substitute, so are we getting the same thing? That is f of a in the first generation is again equal to p, which was same for the previous generation, where f of a in the G naught generation was also equal to p. Again same thing we are getting in the f1 generation. If you do it for the small a in the first, you will get again q, which is again equal to the f of a in the G naught generation will be equal to q, and again in the f1 generation it is equal to q. So this is the proof that the population will be in the equilibrium when it satisfies these conditions. So we have assumed that these conditions are followed by the population. When these conditions are followed, we are seeing that the populations are having the gene and gene frequency which are same from one generation to another generation. So this is with respect to the gene frequency. So similarly, we can also show it for genotypic frequency. Okay. So what are the three possible genotypes which we discussed, that are capital A, capital A small a, and small a, right? So and the frequency of the capital A is p square, frequency of small a is small x q square, and frequency of capital A small a is 2pq. So if these genotypes in the base population is undergoing random mating, for example, what I mean by random mating means like all the genotypes will have the equal opportunity to fuse with, or to fuse with any of the other genotypes, right? So if I just write all of the possible genotypes and make all possible crosses, so here this is what you are going to get. For example, this is the frequency of capital A, and this is the frequency of capital A small a, and this is the frequency of small a. So if we cross this type of individuals, so this is a p square 4, like p to the power of 4, so in the first case, and next it is 2 into p cube into q, and lastly it is p square q square. How we are getting these values? If I just substitute this p square into p square, I'm getting p to the power of 4. Next, this is p square with 2pq, so that would be equal to 2p cube q. So this is the frequency of this possible cross. So if I take all of this into a table where I've given the all possible crosses that were possible from the three different genotypes, and this is the frequency of the individual genotypes. For example, the frequency of this capital A genotype is p to the power of 4, and also it is 2 into p to the power of p cube into q and p square q square. So this is the total frequency of capital A in the population. So if I want to calculate the frequency of capital A in the first generation, what should I do? I should add the individual frequencies. For example, if I add the individual frequency, it would be p to the power of 4 plus 2 to the power of 2 into p to the power of p cube into q plus p square q square. So similarly for all other cases, if I want to calculate the allelic frequencies, or sorry, genotypic frequencies of capital A small a, I have to simply add the individual frequencies, that is 2 into p cube q, 2 into p square q square, and 2 into p square q square, and lastly it is 2 into p q cube. Okay, so similarly if I substitute these values, I will get the frequency of small a small a. So now what we have to prove? So we have to prove that this value is equal to p square. So if we are doing it, or if we are getting it, that means the f of capital A in the G0 generation will be equal to f of capital A in the G1 generation. This is what the equilibrium means, right? That is, the genotypic frequency is remaining constant from generation to generation. It is not changing. Like in the this generation, if it is p square, again in the next generation it will be p square. Again, if it is going for another generation, it will be p square under the certain assumed set of conditions. Okay, so if you just calculate or substitute the values or simplify this, you are going to get the p square here. Okay, so here if I just take this p square as common, since in all of this p square is there, so it would be p square plus 2pq plus q square. So we already know from the Hardy-Weinberg's equation that whenever the population is satisfying the certain conditions which were mentioned in the previous slide, then the p square plus 2pq plus q square is equal to 1, right? So if I substitute that value, so ultimately I'm getting p square in the F1 generation. So this is the exact proof to tell that the population will be in the equilibrium, or the gene and the genotypic frequency will remain constant from generation to generation if it is satisfying certain conditions. I hope this is clear. So let us just discuss about what are these assumptions. So I have discussed about the assumptions in bit briefly in the before slides. Let us just discuss them in detail. First thing, so what we are assuming. So whenever we say that population is in equilibrium, we assume that the population is sufficiently large. So what will be the effect of population size here? What if the size of a population is less? How it is going to affect this Hardy-Weinberg equilibrium? So that we can discuss in the upcoming slides, where when I discussed about the factors influencing this Hardy-Weinberg law or the principle, I will discuss in detail about like how the population size will actually impact or affect this equilibrium or the population. Okay, but for now just know that population should be sufficiently large. Only in that conditions we can apply this Hardy-Weinberg equation. Secondly, it is the organism should be of diploid, where each organisms should have two alleles, like maximum two different alleles at a locus. So this is what it means. Like organism should be diploid, that is it should have maximum two different alleles at a locus, given locus. Okay. Next, so this Hardy-Weinberg law or the principle is purely applied to sexually reproducing organisms. So these are very simple things that or simple points that you have to remember, because this kind of questions you will definitely get in the exams. So remember that it is only applied to sexually reproducing organisms. So why it is only applied to sexually reproducing organisms? Because only in these organisms you see the gametes which are produced from the parents, and these gametes will randomly fuse with the other gametes of the other parent, and then we are going to get the different combination. Since we are going to get the different combinations, we are calculating the gene frequency and the genotypic frequency. But what is happening in the asexual reproduction? Asexual reproduction, the gene and genotypic frequency doesn't change at all. For example, if it is capital A small a in the parent generation, the next generation also it will be capital A small a only, and in the next generation also it will be capital A small a. So that is why this equation is not applicable to the asexually producing organisms. It is only applicable to sexually producing organisms which are having maximum two different alleles at a locus. Okay. Next one. Next most important thing is the population should be completely random mating. That means there should be no control mating in the population. Random mating means each and every individual in the population should have the equal opportunity to fuse with any other individual in a population, and also the gametes which are formed by the individual should have the equal opportunity to survive and fuse with any other gamete. So that is what this random mating mean. And next, so whenever the population or the individuals in the population are undergoing the process of, like cells of individuals are undergoing the process of meiosis, there should be normal meiosis which should take place, and all the gametes which are produced from this meiosis event should equally survive. All of them should survive. So on this condition, the population will obey the Hardy-Weinberg principle or the law. And next, sixth one, all the individuals which are produced should have the equal fitness. Fitness should be equal among the individuals of a population to apply this law. Next, so there should be no overlapping generations. So there should be no overlapping generations within the population if it has to obey, or if we want to apply this principle or the law. So what is this non-overlapping generation? See, whenever you take the populations, there are different stages starting from birth, like maturity stage, and ultimately death. So I'm just briefing it. Okay, so if this is the birth, maturity and death of a certain generation. Okay, so all of these events should be completed before the next generations, the population of the next generation, that is G1 generation, come into the maturity. So before the next generation, the individuals of a next generations are coming into maturity, the older generation should complete its lifespan or life cycle. So this is what non-overlapping means. If I say some population, if I say that certain population is overlapping, that means they are all coinciding. But whenever I say that there is no overlapping generation, that means birth, maturity, death of the previous generation is completed before the next generation is getting matured. Okay, so this has to be satisfied. And lastly, and most importantly, there should not be any evolutionary force that are acting on the population. When these 8 conditions are satisfied, then we can say that the population is obeying Hardy-Weinberg principle or the law. If it is obeying this principle or the law, we can directly use this equation, which is p square plus 2pq plus q square, and with the help of this equation we can calculate the gene frequency and the genotypic frequency without knowing any total number of individuals. Even if we do not know how many number of individuals are there, even without that, if we have this gene frequency, we can calculate the genotypic frequency, and if we are having the genotypic frequency, we can calculate the gene frequency. Without the total number of individuals, we can actually predict it. So what I mean to say is, so in a simple words, see, if we have the genome generation, there are 100 individuals. Let us take, we know that there are 100 individuals. We know f of a is p year and f of small a is q year. Without going for next generation, without calculating the number of individuals, we can theoretically calculate the genotypic frequency based on this data. Before going into the actual breeding, we can do it theoretically if this population obeys Hardy-Weinberg principle or the law. So that is the practical usage of this Hardy-Weinberg principle. Okay, I hope these things are clear. Okay. Okay. Next, we'll see the practical implications or the significance of this Hardy-Weinberg principle. So what are the significance? So that can be of several kinds. So based on which area we are looking into, the uses and the practical implications are different. For example, if we are doing the evolutionary studies, how this Hardy-Weinberg law will be helpful. So if in case we consider that population is and like satisfying the Hardy-Weinberg rules or the principle. So if certain population is satisfying this or obeying this law, that means there is no change in gene frequency and the genotypic frequency over the generation, right? This is what it means. But if there is no change in gene and genotypic frequency, whether there will be chance or scope for evolution in that within that population, whether is there any chance of evolution within the population? No, right? Because the evolution occurs only if there is a certain variation or the change in the frequencies. If there is no change in the frequencies over the generation to generation, then there is no scope of evolution here. So this particular rule or this particular rule will give us the theoretical baseline. That means like we can compare this ideal population with the real population where the all this evolution process are undergoing. See, whatever population you take, there is no population that will obey that will satisfy this Hardy-Weinberg law if we are considering the natural population, right? So this ideal population can be used as a theoretical baseline to track the evolutionary changes that is occurring in the real population. That is how this can be used in the evolutionary studies. And also we can understand about the genetic stability. So if we are knowing like how they are maintaining the gene and genotypic frequency constant from generation to generation. So this is what stability means. We just have certain genes which are very stable, that they are not they are resisting for change from generation to generation, so that we can study from these ideal populations. Okay, so this is in terms of evolutionary studies. Next, in terms of genetic studies, how this Hardy-Weinberg law or the principle is useful. First of all, to study the genetic variation, right? So by the way we are calculating, or because of the calculation of the gene frequency and the genotypic frequency, anyhow we are getting the values for the differences in the alleles, right? Like how, what proportion of the capital A is there and what proportion of small a is there. So that will give us the idea about how much genetic variation is present in the population. And next, we can study about genetic diversity. Just by knowing the genotypic frequency, we can understand like how much diverse the population is. So that is the second important thing. Next, so we can calculate the frequency of diseases here, frequency of diseases which are controlled genetically. For example, let us take a very good example. So if you just read this problem, so they are saying that there is a particular autosomal recessive disorder which is present in 1 in 10,000 individuals. That means, so this disorder, let us take, it might be because of a certain gene A which has two different alleles. They are saying that small a is 1 in 10,000. So what will be the frequency of this recessive genotype? The frequency of these recessive genotypes, even we need not to check for any other data. We do not require any other data to calculate it. Just by seeing this, this is just 1 by 10,000, right? So it would be 0.0001. So that would be the frequency of capital small a, which is equal to q square, right? So let us see what they are asking. So if the population is in Hardy-Weinberg equilibrium, then what will be the frequencies of the two alleles? So they are asking us frequency of capital A and frequency of small a. As I told you previously, even without having a data of like number of individuals, number of alleles, like we can calculate the frequency of the allele if we have a genotypic frequency, and if we have the allelic frequency, we can calculate the genotypic frequency. Now, how can we calculate this allelic frequency? So as we know, they are already saying that it is in the Hardy-Weinberg equilibrium. If it is satisfying this equilibrium, that means if q square is equal to 0.0001, then obviously q will be square root of this value, so which would be 0.01. So q is now 0.01, which is the frequency of small allele. So if we have a frequency of one allele, then we can calculate the frequency of another allele. How? Because we know that p plus q is equal to 1. So p will be obviously equal to 1 minus q, which is 1 minus 0.01. So this will give us the value of p. So this is how simple it is to calculate the allelic frequency of capital A and small a, or p and q, if we are having a genotypic frequency of one of this kind. So this is where it is used. We can use this for studying or predicting the deceased individuals in the population. So I have another examples. So you can clearly see here. So this is another example in which like on how like this law or the principle can be used. So here they are saying that if the frequency of males affected with an X-linked recessive allele in the human population is 0.10, that is 1 in 10. So what will be the expected frequency of the affected females? So they are saying that the frequency of males with the affected X-linked recessive allele is 0.1. That means X and Y, if we take frequency of X and Y, so it is 0.1, right? So this is present in a X, like the gene is or the allele is present in the X chromosomes. And now they are asking us to find out the frequency of this affected female within the population. So this we can do it if we apply this simple Hardy-Weinberg principles. So you can comment your answers on this video, like I will be seeing it and I will be replying each and every answers. Okay, so there is one more question. You can also consider this question, and you can just comment the answers of these two questions. I will be answering or I'll be replying you in the chat box. So that's it for today. So if you like our content, please like, follow, share and subscribe our YouTube channel, and stay tuned for the upcoming videos. I'm planning to just post all of these videos related to the population genetics very soon, before you have your ICAR JRF exam or ICAR PG entrance exam. Okay, so these questions which I have posted here, you can also see that they are asked in the previous year papers, where you can see it was asked in 2023. Okay, so that is why these questions are very much important, and this is how they are going to ask you in the exams. Okay, so if there are any doubts, you can always comment on this comment on this video. And also, like, if you're looking for a complete fully fledged online course for your ICR preparation, ICR entrance exam preparation, that is AIEEA PG entrance exam, which was formerly known as JRF, so you can consider our online master courses which we are conducting, and you can buy or register those courses, where you will be getting fully fledged contents that we are giving, along with the notes, mock tests, doubt clearing sessions, previous year question papers, plus live plus recorded classes. Okay, thank you for listening. We'll meet in the next video.